Okay, good morning. Is that microphone's okay or no? Not on. Oh, how about now? Okay. Can you guys hear me? Okay, good. Yeah.
Could you move the screen down a little bit? It's like
Oh, sorry.
thank you.
All right. So I think last time we stopped this slide, right? Okay. Now, so the next step for glucose oxidation after glycolysis, right? You generate pyruvate under the aerobic condition, right? So the cell has a mitochondria, and also have enough oxygen. So then pyruvate will be further oxidized in the mitochondria, and in the mitochondria, TCA cycle is very, very important for the nutrient complete oxidation, right? So this slide, I think, is very nice to give you the overview of the whole process of nutrient, including glucose and fatty acids and amino acids, complete oxidation, right? So again, acetyl-CoA is a key molecule that connects the three macronutrient oxidation to the TCA cycles. So acetyl-CoA, once it's generated for oxidation of glucose, fatty acids, and amino acids, right, can enter the TCA cycle in the mitochondria and for complete oxidation. And the TCA cycle, we know that is composed of a number of reactions, right? And go through the one TCA cycles and generate ATPs, and particularly and most importantly, the reducing equivalent like FAD, H, and NADH, right? So, and this reducing equivalent is not ATP yet, but it can be entering the the so-called oxidative phosphorylation or electron transport chain to be converted to ATP, right? So there's a whole process you can see, right? Oxidation, acetyl-CoA enter TCA cycles, generating reducing equivalent, NAD, H, and FADH, H, and then further conversion or oxidative phosphorylation, and eventually you generate ATPs. So this is a TCA cycle. So lots of reactions, but you don't need to remember each reaction again. But kind of key things, right? You need to know. First, how many ATPs is generated, right? Or reducing equivalent generated during the TCA cycles. And most importantly, the TCA cycle we mentioned earlier, right? It's called an amphibolic metabolism by pathway. That means this TCA cycle can connect all three macronutrient metabolism, both catabolic and anabolic metabolism. So I just give you an example, like citrate, the oxaloacetate, and the alpha-ketoglutarate, succinate, CoA. These are the most important intermediates or metabolites in the TCA cycles, and this metabolites actually connect to the other nutrient metabolites or the other metabolic pathways. For example, citrate which is generated from acetyl-CoA and oxaloacetate. And citrate can leave the TCA cycle and enter the lipogenic pathways, which is lipid metabolism, right? And for the synthesis of fatty acids. So this is how, glucose metabolism, amino acid metabolism can be connected to lipid metabolism, right? That makes sense. We're going to talk about specific lipogenesis later in the lipid metabolism session. And the other molecules, intermediates, oxaloacetate. This is very important molecule again, and also it's actually enter the gluconeogenic pathway, right? And the alpha ketoglutarate is the molecules or the intermediate that converted from the glutamate, right, the amino acids, and alpha ketoglutarate can enter the TCA cycles and can either be generate the oxaloacetate, right, and enter the gluconeogenesis. So this is how amino acids can be used for gluconeogenesis. This is part of the gluconeogenic pathways, of the group from the using amino acids as a precursors, right? So, and succinate-CoA is also one of the important metabolites that can be converted from the certain amino acids, right, and enter TCA cycle, and can also, go to connect to the gluconeogenic pathways. So this is something that you need to know, the TCA cycles, how important, not only just for the complete oxidation of the nutrients, but also they connected, the different metabolic pathways. So, and try to remember that there's 4 key intermediate that I highlighted here, right, and how they connect to, the different metabolic pathways and fatty acid synthesis and the gluconeogenesis. And the ATP production, right, from the calculation of ATP production. So this is also, I think, is a good exercise to help you to understand, the whole process or metabolic pathway of a glucose oxidation metabolism. Okay, this is the last slide for this. So if we already calculated, right, the ATP production from glycolysis, the one glucose molecules, if it's completely oxidized, for example, in the brain, right, the glycolysis is only the part of the oxidation process, right? Then the pyruvate being further oxidized in the TCA cycles, and so when you calculate the the ATPs, glycolysis pathways, one glucose. If you still remember how many ATPs can be produced from one glucose molecules through the glycolysis pathway? No, I mean in the aerobic cells. 6 to 8, right? 6 to 8. And then how many acetyl-CoA can be generated from one molecule of glucose in oxidation? 2, yes. So then 2 acetyl-CoA molecules will enter the TC cycle for complete oxidation, right? Now, how many the ATPs can be produced or generated once finish the one cycle of the TCA, TCA cycle. We have not talked about it yet, right? And also I wanted to, don't forget actually one more step in the mitochondria, that is pyruvate, right? Converted to acetyl-CoA. And this step also produces NADH, you remember, right? So, and the two pyruvate generated from one glucose oxidation, right, and enter the mitochondria. So you should expect to have two NADH, right, produced from the reaction, pyruvate converted to acetyl-CoA. So two NADH generated in this reaction, and then acetyl-CoA enter the TC cycle. So if we go back to the TC cycle, you can see that actually a one ATP molecule is produced in the TC cycles, right, at the substrate phosphorylation level. And how many NADH? 1, 2, 3, right? 3 NADH and produced from TC cycles, and 1 FADH is produced, right? So again, the one TC cycle can produce this 1 ATP, 1 FADH, and 3 NADH molecule, right? So then you're thinking about, you calculated, how many total of ATPs can be produced when finished the one TCA cycle. One NADH, once it goes through the electron transport chain, will produce 3 ATPs, right? And FADH will produce 2 ATPs. So 3 plus 2 will be 5, and plus 1 ATP at the substrate phosphorylation level. So 6 ATPs, one cycle of TCA cycles, right? That means again, one molecule of acetyl-CoA, once complete the one TCA cycles, is able to produce 6 ATPs. And we know 2 acetyl-CoA actually enter the TCA cycles, right? So the total will be 12 ATPs generated. Once one molecule of glucose oxidized, complete oxidized, right? So the total ATP will be Y24. Do you guys have an idea why it's 24? Sorry, I did that. I messed up. I think I did the wrong calculation. The 3 ATPs from 1 NADH, right? And we have a 3 NADH, so actually 9. Sorry, 9, and then plus the 2, plus the 1 ATP, and then plus 2 from FADH. So actually 12. Sorry, my math is kind of bad. So again, one molecule of acetyl-CoA enter the TCA cycles and complete the TCA cycles and generating 12 ATPs, right? And 2 acetyl-CoA usually generated from oxidation of 1 glucose molecules. So we'll need to times 2, right? By 2. So total actually 24 ATPs generated from TC cycles, right? And 6 basically is because, from this pyruvate dehydrogenase reaction, pyruvate converted to acetyl-CoA, right? 1 NADH basically generates 3 ADPs and times 2, 6. Yes, questions, I saw. Yeah.
Can you explain why you guys multiply by 2?
Oh, why we multiply by 2? Do you guys have any idea why? Who can? Anybody? Yes. Right. One glucose molecules break down into 2 pyruvate, right? And 2 pyruvate molecules converted to 2 acetyl-CoA. And so basically you have a 2 acetyl-CoA enter TC cycle. Right? Does that make sense? Okay. So this just, help you to thinking through that, start from glucose and glycolysis, pyruvate enter TC cycles, and pyruvate dehydrogenase reaction converts pyruvate to acetyl-CoA, right? And acetyl-CoA enter the TC cycle and complete the oxidation. Okay, so let's get the okay, now we're going to talk about gluconeogenesis and exoprosis and cataprosis. Let's just glycolysis again, let's just review it. And so glycolysis again occurs in the cytosol, right? And basically it's a reaction from from glucose and to pyruvate or lactate, depend on again whether the cell is under the aerobic condition or anaerobic condition, right? And aerobic condition, the final product of a glycolytic pathway will be pyruvate, right? And pyruvate can further oxidize in the mitochondria. And if cell is under the anaerobic condition, and pyruvate will be converted to lactate. So lactate is the final product of glycolysis in the anaerobic cells. And aerobic condition, again I just mentioned, right? Glycolysis occurs in the cytosol and generate pyruvate, and which enter the mitochondria for further oxidation to acetyl-CoA and enter the TC cycles and will complete oxidation and generate CO2 and water, of course most importantly ATPs, and reducing equivalent. And this reducing equivalent then need further to be converted to ATP through the oxidative phosphorylation or electron transport chain, right? And this process, oxidation actually requires oxygen and also in the mitochondria. So that's for the glycolysis and TC cycle, the glucometabolism. Now I just wanted to further talk about the electron transport chain or respiratory. We have several different names, respiratory chain or electron transport chain, oxidative phosphorylation, right? All the same thing. So the electron transport chain basically is a complex, right? It consists of the 4 enzymatic complex, right? Complex 1, 2, 3, and 4. So each complex actually has a multiple enzymes, right? for example, complex 1 is called NADH dehydrogenase, but not just this, just only have this enzyme. They actually, other several enzymes work together, right, to compete, mainly the dehydrogenation function. But we're not going, to know all the details, what kind of enzymes in each complex, right? But just, it's not just the one enzyme, right? Several enzymes work together. And an important thing that you need to know that complex 1, 3, and 4 actually are the proton pumps. So that means during the electron transport activities, the proton can be pumped out of the inner mitochondrial membrane, right? So the meantime, the proton gradient, concentration gradient can be generated across the inner mitochondrial membrane. And this proton gradient concentration contains chemical energies, right? And this chemical energies can be used for synthesis ATP, right? So complex 1, 3, and 4 has the proton pump function, right? So that means electron, if transported to complex 1, 3, and 4, and this 3 places site can generate the proton gradient, right? And one proton gradient at one side, normally that energy is enough for synthesis of 1 ATP molecules, right? And also ATP synthesis, somebody also called complex 5, but it's fine. So ATP synthesis is also an ATP-dependent proton pump, and this ATP synthesis actually pump the proton the opposite from from outside of inner mitochondrial membrane to inside the mitochondria. So that actually you reduce the proton concentration across inner membrane. So then you can release the energy and transfer the energy to ADP molecules and forming ATP, right? And just you probably know that, right? And oxidative reduction reaction. Sometimes oxidation reaction, not just the reactions that involve oxygen, but sometimes the electron transport or the proton transport reaction also called oxidation and reduction. So oxidation reaction means that a chemical substance, if chemical substance take on oxygen or loses the electrons. So here we specifically, oxidation reaction in this electron transport chain specifically refers to somehow the loss of the electron. So this kind of reaction is called oxidation reaction, right? And reduction is that a chemical substance can give up the oxygen or take on the electron. Here, specifically, we again talking about the substance that take on the electron. Electron, this kind of reaction called reduction, right? Then electron can be transported through this 4 complex from the NADH or FADH molecules, right? And generate proton gradients, concentration, and and then transport its energy in the proton gradient concentration to the ADP and synthesis for the synthesis of ATP, right? Does that make sense, right? So here give you a clear picture of a diagram. Give a clear picture of 4 complexes where they are located. So this is the inner mitochondrial membrane, right? It's the inside of mitochondrial matrix, and this the outside is also called an intermembrane space between inner and outer mitochondrial membrane. And complex 1, 4, 3, and 4, and also ATP synthesis, actually are located on the inner mitochondrial membrane, right? And complex 2 actually located inside of inner mitochondrial membrane or mitochondrial matrix. So that's why complex 1, 3, and 4, and also 5 is ATP synthesis. They are the proton pump, so they be able to pump proton out of inner mitochondrial membrane and generating the proton gradient, by being able to generate a proton gradient across the inner mitochondrial membrane. But the complex 2, because they are located inside mitochondria, right? So the complex 2 does not have this proton pump function. So that maybe help us to understand why NADH can generate 3 ATPs and FADH only 2 ATPs. You can see NADH usually transport electron, proton to complex 1, right? And then 3 and 4 and 5. So, and as I mentioned, each complex that generated proton gradient is enough for 1 ATP synthesis, right? The energy is enough for 1 ATP. So that's why NADH transport electrons through electron transport chain 1, 3, and 4 be able to generate 3 ATP. Does that make sense, right? And FADH usually transport its electron to the complex 2, and then complex 3 and 4, and bypass complex 1. So that's the reason why FADH is only able to generate the 2 ATP molecules, right? Any questions? Right. Now next we're going to talk about the transporter shuttle system. If you still remember, we mentioned the NADH that produced from the glycolysis in the cytosol, right? Sometimes, or in some type of cells, and produce 2 ATPs, and other type of cells produce 3 ATPs. Again, depend on the shuttle system that transport the NADH from cytosol into the mitochondria. Remember this, right, concept. So here I just wanted to tell you this 2 shuttle system that help transport NADH from cytosol into the mitochondria. So NADH is not a membrane permeable, so it itself cannot be easily or freely enter the mitochondrial membrane, and requires a transporter system to help NADH enter the mitochondria. So there are 2 shuttle systems. The first one is called glycerol 3-phosphate shuttle. And see, this is the outer mitochondrial membrane and inner mitochondrial membrane. So the NADH here in the cytosol basically is from glycolysis, right? Again, cannot pass through the outer mitochondrial membrane, so it needs this shuttle system. So basically, the proton can be transferred from NADH to dihydroxyacetone phosphate, and generating glycerol 3-phosphate, right? And this glycerol phosphate carries the protons from NADH, right? And glycerol 3-phosphate is permeable to outer mitochondrial membrane, so it can easily pass through the outer membrane and enter the interface, right, the intermembrane space. And then the glycerol 3-phosphate will give of the proton, which initially from NADH, right, and to the FAD in the inner mitochondrial membrane. And FAD will receive the protons from glycerol phosphate and then convert it to FADH. So, and it is this FADH that enter the electron transport chain for ATP production, right? So this shuttle system will, if NADH from cytosol enter the mitochondria through glycerol 3-phosphate, shuttle system will only be able to generate 2 ATPs in the mitochondria. Does that make sense, right? Because it converted actually, to the FAD. So FAD enter the electron transport chain, like complex 2 and 3 and 4, so on. So only generate 2 ATP. Does that make sense? All right. Okay. So and the other shuttle system is called a Mealy S-body shuttle, and it's pretty complicated. I'm not going to go to the detail, but similarly mechanism, the NADH produced from glycolysis in the cytosol give its proton to Mealy, to oxaloacetate, and oxaloacetate receiving proton from NADH and convert it to Mealy. And Mealy be able to pass through inner mitochondrial membrane and then give the proton to NAD and forming NADH. So eventually NADH in the inner mitochondrial membrane then enter the electron transport chain. So this shuttle system, because of the the NADH eventually enter the electron transport chain, right? So be able to produce 3 ATPs. So again, depend on, and this shuttle system not always present in the same cells. Some type of cells may be predominantly, mainly a spotted shuttle and not much glycerol 3-phosphate, and the other type of cells with predominant shuttle system is the glycerol 3-phosphate, right? So that's why depend on what type of cells, and that determine whether NADH from cytosol from glycolysis can generate 3 or 2 ATPs. All right, does that make sense? So that explains, why sometimes, NADH from cytosol generate 2 or sometimes 3 ATPs. And I just wanted to remind you again, the NADH produced in the TCA cycle, because it's already in the mitochondria, right? So will always generate 3 ATPs, right? So only NADH that produce in the cytosol and depend on the shuttle system whether they produce 3 or 2 ATPs, right? Okay, now I think we now finished one metabolic pathway like glycolysis, TCA cycle, complete oxidation, and how reducing equivalent that generated from these pathways produce ATPs. Now let's talk about a different metabolic pathway of glucose metabolism, gluconeogenesis, right? Gluconeogenesis is the metabolic pathway that can synthesize glucose from non-carbohydrate precursors, right? And the purpose of the gluconeogenesis in our body, that's the gluconeogenesis, is to maintain blood glucose level within the normal range. And so what do you think? It occurs primarily in which tissues? We talked about earlier, right? Liver and kidney. So it primarily occurs in liver and kidney, right? And it occurs primarily in which cell, cellular compartment, which part of a cell? Mitochondria, or cytosol, or thinking about how cells completely oxidize glucose in which part of cellular compartment, right? And also cytosol, right? The first part of glycolysis is cytosol, right? And then the second part of the complete oxidation, mitochondria. So involve both cytosol and mitochondria for complete oxidation of glucose. And gluconeogenesis is simply just, say kind of opposite, right? You produce the glucose from non-glucose sugar. So it should be also involved in both, right? Like mitochondria and cytosol, right? And in which physiological conditions would you expect like gluconeogenesis would be? Fasting, yeah, very good. So fasting. Now this is something that you need to remember, right? The common precursors for gluconeogenesis, what molecules that can be used for synthesis of glucose. In general, it's non-sugar, right? Non-sugar molecules. So like pyruvate, lactate, glycerol, propionate, and also several, or more than several, right? Many gluconeogenic amino acids can also be used for synthesis of glucose. For example, any type of amino acids, if it can be converted to pyruvate, like alanine, serine, glycine, threonine, cysteine, or any type of amino acid that can be converted to oxaloacetate, like aspartate, right, or alpha-ketoglutarate, glumine, right, and the other amino acids. All these amino acids, as long as they are able to convert it, pyruvate, oxaloacetate, alpha-ketoglutarate, right, enter the TCA cycles, then this amino acids be able to use for synthesis of glucose, right? So metabolic reactions in the gluconeogenesis is not just a simply opposite of a glycolysis pathway, right? Most of the glycolytic pathway and complete oxidation of glucose is reversible. That means you can go the opposite direction. But three reactions in the key reaction in the glycolysis glucose oxidation pathway is irreversible. So that means you cannot go the opposite direction to complete the gluconeogenesis pathway, right? So these three reactions, including one, is glucose, oxidized to glucose 6-phosphate by glucokinase, hexokinase. This is the first key reaction, right, in glycolytic pathway. Still remember, and then the second is the key reaction in the glycolysis is the fructose 6-phosphate converts to fructose 1,6 biphosphate and by this key enzyme, phosphofructose 1 kinase. And the last one is the phosphoenolpyruvate converted to pyruvate by pyruvate kinase. And these three reactions cannot go the opposite direction by the same enzyme. What I'm saying is, the cannot be irreversible. That means cannot be go to the opposite direction by the same enzyme. So this key enzyme only catalyze the reaction go to this direction, right? Cannot go opposite direction. So in the gluconeogenesis, we need a different sets of enzyme to catalyze opposite reaction for this three key reaction, right? Does that make sense? So if I just do the opposite reaction, for example, or I'll go to the top one. Pyruvate cannot be converted to phosphoenolpyruvate by pyruvate kinase, right? But we need two enzymes called pyruvate carboxylase and phosphoenolpyruvate carboxylase, and these two enzymes are the enzymes that be able to catalyze the conversion of pyruvate to phosphoenolpyruvate, so go to the opposite direction, right, in the gluconeogenesis. So these two enzymes are the part of the key enzyme in gluconeogenesis that you need to remember, right? And in the middle one is, the fructose 1,6 biphosphatase, and this enzyme catalyzes the opposite direction, the reaction from fructose 1,6 biphosphate and converts to fructose 6 phosphate, right? So fructose 1,6 biphosphatase is the key enzyme in gluconeogenesis, right? And phosphofructose 1 kinase is a key enzyme in glycolysis, right? So two different sets of enzymes in glycolysis and versus gluconeogenesis. And similarly for the last step, glucose 6 phosphate converts to free glucose, and this reaction is by glucose 6 phosphatase. You already know this enzyme, remember, right? And we explain why muscle cells cannot release glucose because of lack of this enzyme, right? So this is one of the key enzymes in the gluconeogenesis, right? So these three are actually four because this, two reaction, right, make the conversion. So four key enzymes in the gluconeogenesis and catalyze, this three key reversible reactions in the gluconeogenesis, right? All right, let's take some break, right? Maybe 8 minutes or so. All right. Now let's go to the details. The reactions, just give you an overview, but again, you just need to remember the key reactions and enzymes in gluconeogenesis. Okay? So basically, this is actually the whole process of the glucose complete oxidation, right? From glucose glycolysis pathways and generating the, let me see where is it. Okay, so you know the complete oxidation, right? Oh, sorry, actually from so the gluconeogenic pathway is actually go the opposite direction, right? And this is the mitochondria, and this part represent the cytosol. So gluconeogenesis, we know they have a variety of precursors, right? And the pathway is actually a little bit different, depending on the cell use which type of precursors for gluconeogenesis. So if lactate or pyruvate as the precursors, and it will involve all 4 reactions in the gluconeogenesis, the 4 bypass reaction, I would say, right? Bypass reactions. 2 reactions in the mitochondria and the other 2 reactions in the cytosol. So let's see the lactate or pyruvate as the precursors, right? The 2 reactions in the mitochondria include, one is the conversion of pyruvate to oxaloacetate by pyruvate carboxylase, and the second reaction in mitochondria is the conversion of oxaloacetate to phosphoenolpyruvate or PEP, and involve these 2 reaction enzymes, right, in the mitochondria. Let me just go to the details. Next step. So in general, just try to remember the 2 reactions occur in the mitochondria and the other 2 reactions in the cytosol, and different precursors. They probably use different pathways to synthesize glucose, right? And lactate and pyruvate, if these 2 molecules as the precursor that involve the complete the 4 reactions bypass, 4 bypass reactions, right? And if glutamate as a precursors and only involves actually 3 reactions, one in the mitochondria and the other 2 in the cytosol, and if glycerol, we use the glycerol as a precursor, it only involve the 2 reactions in the cytosol, right? It does not involve in the mitochondria. So in some cases, for example, somehow, pathological conditions, the mitochondria may have some problems, cannot function efficiently. So pyruvate or lactate may not be able to efficiently use for gluconeogenesis, but glycerol will be able to use to, still be able to use to generate the glucose, right? And since this pathways does not involve the mitochondria, right? So this some details, and you can see, and I just already mentioned, right, 2 reactions in the mitochondria, and pyruvate and lactate, right, as the precursors that involve these 2 reactions. And glutamate, the 2 things that you need to know, that glutamate only involves the 3 reactions, right? One in the mitochondria, and the other 2 in the cytosol. And the other thing is that glutamate also, the gluconeogenesis from glutamate is also involved the TCA cycle, right? Glutamate, if you still remember, can be converted alpha ketoglutarate, which is a key intermediates, right, in the TCA cycle I mentioned, try asking you to remember. So after ketoglutarate and goes through the several reactions in the TCA cycle, then converts to oxalic acetate, and oxalic acetate leaves the TCA cycles and be converted to PAP, right? So only involve this reaction, the catalyzed by PAP-CK or phosphoenolpyruvate carboxykinase, right? Only this reaction is needed for gluconeogenesis from glutamate. And cholesterol, again, the pathway, gluconeogenic pathway from cholesterol is kind of relatively short, right? It only involves 2 reactions in the cytosol, and that is fructose 1,6 by phosphatase and glucose 6 phosphatase, right? The last 2 reactions that involve in the gluconeogenesis from glycerol in the cytosol, right? And this, I think it's just kind of detailed reactions, fructose 6 phosphate to fructose 1,6 biphosphate, This actually kind of connect, glycolysis and gluconeogenesis together to show you the 2 different sets of enzymes, right? And catalyze the opposite reaction in the opposite direction. So the right side part is actually the glycolysis reaction in the glycolysis, right? And the enzyme that involves glycolysis is the phosphofructose kinase, right? Catalyze the conversion of fructose 6 phosphate to fructose 6,1,6 biphosphate. And the left side, and the reaction from the bottom to the top is the direction, the gluconeogenesis direction, and this is a different enzyme, right? Catalyzes the reaction, fructose 6,1,6 biphosphatase. And similarly for the last reactions in the gluconeogenesis, but first reaction in the glycolysis, right? Glucose conversion to glucose 6 phosphate. So this reaction in the 2 directions regulated by 2 different set of enzymes in 2 different metabolic pathways, right? So that we're going to talk about later, how gluconeogenesis and glycolysis is being interregulated, because glycolysis and gluconeogenesis, their activities in the different, the activation should be in the different physiological condition, right? Glycolysis should be more active in the fat condition, and gluconeogenesis should be in the fasting condition, right? So we can activate both pathway activities in the one condition, the physiological condition, right? So we need to activate one and then reduce the other one, pathway activities. So these 2 sets of enzymes, they are oppositely regulated by some hormones, for example, insulin. So insulin, if insulin is increase the glycolysis basically, right, and decrease gluconeogenesis. So insulin basically upregulates the glycolytic enzyme like glucokinase or hexokinase, but inhibits the gluconeogenic enzymes, right? So, so in that way that, one pathway can be upregulated, other way can be downregulated under certain physiological conditions, right? We will talk about this more detailed later. Now I'm going to mention the two very important cycles for doing, the gluconeogenesis. One is called Cori cycles, and the specific gluconeogenic pathways. That involves the synthesis of glucose from lactate. And Cori cycles involve three tissue cells. Liver, of course, is the place that gluconeogenesis occurs, right? And muscle and red blood cells are the two tissue cells that provide the precursors, which is lactate, right? So lactate, we know that because of red blood cells usually don't have a mitochondria, always go to the partial oxidation glucose and produce a lot of lactate, right? So it's a major sources of lactate, and muscle can also produce a lot of lactate. So both cell tissues contribute lots of lactate, and for liver to synthesize glucose. So the process of cycles we call the Cori cycle, right? So Cori cycle, the purpose of Cori cycle is to do the gluconeogenesis and provide glucose to the circulation. And three tissue cells involved, and the precursors that involve this Cori cycles is lactate, right? And the other cycle is called alanine cycle. So alanine cycle, the muscle and liver is involved in alanine cycles. So alanine cycle basically is muscle regulate, break down the proteins, right, during the fasting condition, and provide a alanine. So alanine can transport it to the liver and as the precursors of gluconeogenesis for glucose production. And of course, they also involve like transamination and deamination in these two tissues, right? And similarly, the purpose of alanine cycle is also, to provide glucose through the gluconeogenesis, and involve the alanine as the precursors of gluconeogenesis. And this slide, I think we mentioned earlier, if glutamate as a precursors only involved three bypass reaction in gluconeogenesis, right? One in the mitochondria and the other in the cytosol. So the bypass basically is the pyruvate, the carboxylase reactions, this reaction. And of course it involved the TC cycle. There are two things you want to know. Glutamate as a precursor that involve TC cycles, and also only involve the three gluconeogenic bypass reaction for gluconeogenesis. So this is again the TC cycles. I just want to emphasize this part. This actually is part of a gluconeogenic pathways when amino acids as the precursors, right? Like glutamate is metabolized to alpha ketoglutarate and enter the TC cycles and go through several reactions and convert to oxaloacetate in TC cycle, and then this molecules is converted to, if you still remember, the PEP, right, and enter the gluconeogenic pathway. So this is, the glutamate that involve TCA cycles for gluconeogenesis. And glycerol, and just enlarge it a little bit of the pathway so you can see more clearly. The glycerol that only involve this last two bypass reactions in the cytosol, right? It's used as a precursor. All right, so enzyme that utilize in the gluconeogenesis. So gluconeogenesis, this reactions or pathway that is kind of anabolic pathway, that right? So you need energy to do all these activities. So basically, energy is used from the fatty acid oxidation or partial amino acid oxidation, which makes sense, right? During the fasting condition, we always want to spare glucose, and for use by brain, right, and red blood cells. So liver is a place that synthesizes glucose. That's gluconeogenesis. So we don't want to use a lot of glucose, right? ATP, ATP from glucose for gluconeogenesis in liver, and we wanted to spare glucose and release the circulation and pick up by brain and red blood cells. Those tissue cells actually heavily rely on the glucose for energy, right? So that makes sense. I mean, gluconeogenesis, the energy mostly from fatty acids oxidation and also amino acids oxidation, right? So this is the last slide I'm going to talk today. Let's just put everything together and think, the gluconeogenesis during the starvation, like that particularly involve the liver and the muscle. I think I have not talked about the kidney yet. I mean, kidney also is important site, right, for the gluconeogenesis, but later. So far that we covered, the liver and muscle, we talk about 2 cycles, right, for gluconeogenesis. And main purpose is during starvation, we wanted to maintain the blood glucose level. The main purpose is for the use of energy by brain and red blood cells, right? And liver is the main place that synthesizes glucose from precursors. And 2 cycles, choline cycle and alanine cycle, just involve the 2 precursors, right? Lactate basically is from muscle oxidation of glucose, and also from red blood cell, right? And alanine is from muscle and provide precursors for liver to synthesize glucose. And of course, some other precursors like glycerol and amino acids, right? We didn't show here, but just let you know, it also be able to use for gluconeogenesis in the liver, glycerol and amino acids. All right. Any questions so far? All right. So let's have a quiz.